हेलो।
भाई और बड़ा मुझसे बड़ा कुछ है काम है। नाम को एक काम से मतलब आगे में नाम है आधा ऊपर लिख दिया आधा नीचे लिख दिया तो साइन कर मैं कैसा लग रहा है छोटा वाला छोटे वाले में बनाइए आप ऐसी हाँ, ऐसी छोटी में ये बड़ी है ये हैंडल पे इसका है
अरे गिरिराज जी, आपका आपका माइक ऑन है, जरा बंद करिए इसको। डॉक्टर अरविंदा साहब ने ज्वाइन कर लिया है, आप उन्हें कोच बनाइए। रोहित जी, क्या हुआ? पार्टिसिपेंट्स पीपीटी के साथ रेडी नहीं है क्या?
नहीं शायद सर, अभी तो नहीं, शायद कुछ तो ग्लिच आ रहा है।
अच्छा, जिनका है उनका ही स्टार्ट कराओ आप, जो है अभी।
हाँ सर, आप एक बार ग्रुप में मैसेज ड्रॉप कर दीजिए सर।
मैं ग्रुप जब सबका शेड्यूल दिया गया है तो इस तरीके से थोड़ा ना, तो हम कराएंगे ही नहीं फिर, और क्या है, हम उनको लिखे दे रहे हैं अभी।
Madam Preeti, you can start. अंकुर सर, प्रीति मैम रेज कर रही है हैंड।
हाँ तो प्रीति जी को, हाँ बन गई है प्रीति, एच यू इज द को होस्ट, उनको बोलो वो प्रेजेंट करें। हाँ प्रीति, आप प्रेजेंट करो।
गुड मॉर्निंग सर।
हाँ गुड मॉर्निंग। पीपीटी शेयर करो अपनी।
या श्योर सर।
हम्म।
Yeah, madam. Am I audible to you? The presentations can be started, please.
यस मैडम।
सर विजिबल है?
Yeah, is it visible, madam? Patient, global.
Yes, visible, ma'am.
Good morning, everyone. Today I am here to present my presentation on the topic recent advances in production and production of crop under the open and production condition. I'm audible?
हाँ हाँ आप audible हैं।
In this, we discuss about agripreneurship is cited as one of the most important drives of economic development. During the economic crisis, when the development was negative, the importance of polyhouses for the development of agripreneurship increased. Polyhouse agripreneurship represents a transformative force in modern agriculture, offering a sustainable technological advances approach to crop cultivating by harnessing the power of control environment, precious agriculture, and entrepreneurs with polyhouse farming holds the promise of enhanced food security, promoting environmental sustainability and forecasting economical prospects. There are different type of polyhouses in use in Haryana and all the states of India. Natural ventilated pads and fan polyhouse, double layered polyhouse, high tech polyhouses. And the objective are the study aim to clarify the role of polyhouse as a farmer in agricultural development in India. Basic of classification of greenhouse based on their shape, utility, constructions, and their material. In the shape, learn to type of greenhouse, even span type of greenhouse, uneven span type of greenhouse, ridged greenhouse, interlocking, and ground to ground greenhouses. Utility greenhouses for active heating and greenhousing for active cooling according to their crops. Wooden frame structure, pipe frame, thresh frame structure, and based on their material, grass, fibers, plain sheets, and plastic films, UV stabilized, and net houses, high cost of greenhouse, low cost greenhouse. Role of agripreneurship development of Indian economy. Agripreneurship serves as a dynamic engine of economic growth, unlocking the latent potential of India agriculture sector through innovation, enterprise, and value additions. By harnessing cutting edges technologies, modern families practices, and business accounts, agripreneurship drives productivity enhancements, cost efficiency, and the market diversion, thereby enhancing the competitiveness and profitability of India agriculture. For precision farming and organic cultivation, for agro-tech startups and food processing ventures, agripreneurship spans and spectrum of initiatives aimed revolutions the way of produced proceeds contributions. Greenhouse farming states in Haryana, Rajasthan, Maharashtra, Karnataka, all the states used grow crops by polyhouse. Top 5 states greenhouse horticulture states are first in Maharashtra, second Haryana, Karnataka, then fourth Gujarat, and fifth Tamil Nadu. Government also give assistance to the farmer for polyhouse. Low area cost 260 meter square, size of polyhouse, total cost structure, subsidies, and farmer shares. Conclusion. The percentage of total variable cost over 65% of two crop in each condition, so highly higher condition, outdoor condition, while the percentage fixed cost, the cost low cost, higher the crop were grown outside. On other hand, higher yield were observed in polyhouse cultivation than the outdoor conditions. For all crops, net yields increase more than double when grown polyhouse. Yield per rupees were higher than value rupees 2.27, 2.21 respectively. Summer squashes and tomatoes were undergrown in polyhouse condition compared to 2.11, 1.96 and its close outdoor conditions. Thank you.
Yeah, good presentation, Preeti. Could you throw some light on the different crops which are profitably grown under the polyhouse conditions?
Ma'am, summer squash and tomatoes.
Summer squash and tomatoes. Okay. So, what is the, advantage of polyhouse as compared to the outside cultivation with respect to season?
Ma'am, I'm not audible to you.
Yeah, you are audible. What is the advantage that you have under protected cultivation of these crops as compared to outside environment?
मैम मैंने विदाउट पेस्टिसाइड्स क्रॉप ग्रोन की है क्या कहते हैं पॉली हाउस के अंदर कंपेरेड टू आउटडोर के अंदर इंसेक्ट्स भी होते हैं बर्ड्स भी होते हैं जो उस क्रॉप्स को कुछ खराब कर रही है या हल्के हल्के कुछ डिजीज आ रहे हैं आउटसाइड की क्रॉप में बट कंपेरेड टू आउटडोर एंड इंडोर के अंदर इंसेक्ट्स भी नहीं है पेस्टिसाइड्स भी नहीं है एंड क्रॉप प्रॉपरली ग्रो हो रही है आउटडोर के अंदर मतलब उनको वहां पे स्टे करने के लिए भी, इजीली स्टैंड करने के लिए भी टमाटोज की जो प्लांट होता है उसको इजीली कंफर्टेबल नहीं हो रहा, बट पॉली हाउस के अंदर वो इजीली कंफर्टेबली स्टे कर रहे हैं और उसके अंदर इजीली स्टैंड कर रहे हैं। प्रॉपर अमाउंट ऑफ वाटर भी कम है अंदर, और आउटडोर में वाटरिंग भी ज्यादा है क्योंकि गर्मी की वजह से बाहर पानी भी ज्यादा चाहिए, अंदर कम चाहिए।
Yeah, what are the tests that you have told which could be, I mean, less incidence in polyhouse condition in these crops?
प्लीज रिपीट मैम।
What are the pests that were to a lower incidence in protected cultivation? You told the pest disease was less.
मैम इन आउटसाइड वाइट फंगस आ रही है समर स्पेशस में,
Okay.
एंड टमाटोज भी खराब हो रहे हैं।
So, what percentage of the farmers are following this protected cultivation? कितना percentage of farmers are following, do you think?
रोहित जी, भाटेका मैडम को को होस्ट बनाइए आप।
Yes, Preeti?
मैम नॉट ऑडिबल।
नहीं. कितना
Dr.
percentage of
Bhanureka?
farmers are following this protected cultivation? How many farmers are following this? Percentage of the farmers following the protected cultivation.
Okay. Dr. Preeti, am I audible to you? I am the chairman here, Dr. Narayan Reddy. Dr. Preeti?
प्रीति, आवाज आ रही है आपको?
यस सर,
आप
यस
सुन रहे हैं
सर,
ना? मुझे सुन रहे हैं आप?
यस सर, यस सर।
Yeah, you have done, your presentation was good, but at the same time, you have to touch the weather parameters. Say meteorological indices you have not given. What is that you have controlled? Especially when you are presenting a good paper on polyhouses, shade nets, or controlled atmospheric storage conditions, or raising conditions, growing conditions. Under these situations, is it your original work or you have reviewed it?
Is reality work, sir.
Is it?
Real work. My work is also on polyhouse and open field.
And you have not given the weather parameters. See, what is the ambient temperature or humidity? Then what is that under controlled conditions? How much temperature or other?
Humidity is more in polyhouse than comparative to outside.
You should have given, not, don't say more or less. You should have given say 1 or 2 slides. That should have given ample response. When Dr. Bhanureka is asking, you should have answered many of the questions. Had you given 1 or 2 tables, is it not?
Yes, sir.
You have the data? You have
yes,
the data,
sir,
no?
but yes,
No,
sir.
right now I know you have not presented here. You do have the data,
Yes,
no?
sir.
That speaks
Yes, sir.
volumes. That speaks volumes. Dr. Preeti, thank you very much.
Yes, sir.
We go to the next speaker.
Yeah, good Preeti, good presentation.
Yeah.
Okay, thank you sir. Proceed to
Yes.
next. Dr. Rohit, next के लिए बत बोलिए।
Yeah. Now, Dr. Arvinda, you want to speak something?
Yes, sir.
Please go ahead.
Morning to all the
दो
Good
सिस्टम
morning
से
to-
ज्वाइन किया है मैडम क्या? थोड़ा सा वॉइस आपका इको
coordinators and the participants. This is Dr. Aravinda, Associate Professor from the Department of Plant Biochemistry in Sri Krishna Deva Raya College of Horticultural Sciences, Anantapur. As one of the coordinators of the 2024 International Conference, on behalf of Sri Krishna Deva Raya College, I would like to introduce Dr. N. Narayan Reddy, sir, the Associate Dean of the college. Dr. N. Narayan Reddy did his MSc AG Horticulture from GPBAUT Pantnagar and PhD from IARA New Delhi. He started his career as a scientist at ICR, Indian Institute of Horticultural Research, Bangalore. For 7+ years ago, worked as principal scientist and head in
मैडम
ICR.
स्क्रीन पे आपका पेपर विजिबल हो रहा है जिससे आप पढ़ रहे हैं, देखिए आपका फेस नहीं दिख रहा है।
of horticultural crops before superannuating as principal scientist at ICAR, Central Research Institute for Dryland Horticulture. He is an expert in tropical and subtropical fruit crops, vegetable crops, plantation crops, cropping systems involving horticulture-based production systems, etc. He contemplated and contributed to the climate resilient horticulture practices under a mega project entitled National Innovations in Climate Resilient Agriculture in India. He is an expert member for various national and international bodies, INDIA, GAP, NHP, ABMIP, AB Agros, SRLS of NATP, Land Degradation and Sustainable Rural Livelihoods, Indo-Israel Project, and so on. He handled several research as well as developmental programs as a leader or core team member of NATP, NAIP, NICRA, RSAD, AICRPDA, AICRPAM, and generated a fund of rupees 111 crores. Established linkages at national and international level, handled institutional, sponsored, contractual, and consultancy products. He organized 11 summer and winter schools, refresher courses, symposia, conferences, introduced new courses and guided students for UG, PG, and doctoral programs. He published 53 papers in peer-reviewed journals, authored 4 books, 46 book chapters and manuals, 77 conference proceeding papers, 45 popular articles, and 15 bulletins and policy briefs. He is a freelance consultant to CSR projects of Tatas and other grooming entrepreneurs, industry groups, and multinational LLPs offering end-to-end solutions involving horticultural commodities or components with the systems approach. He brought laurels to the institutions wherever he worked, apart from getting many personal awards. Now I request you, Dr. Narayan Reddy sir, to deliver few words.
Now that good number of papers are there, and I request the next speaker, contemplating the salient points of their own work or whatever they have collated, you kindly go ahead. Who is the next speaker, please?
Okay, thank you sir, thank you. Next को प्रोसीड करिए. बहुत अच्छा इंट्रोडक्शन रहा आपका, बहुत बेहतरीन सर.
Okay, so Dr. M. Bilashini, are you there?
Yes, sir.
Okay,
Yes,
we
sir.
can
I'm
start
here,
with-
sir.
Okay.
Yes, sir. Sir, my slide is visible, sir.
Yes, ma'am.
Yes, madam.
Good afternoon to
Go ahead,
respected.
Dr. Bilachun.
Yes, sir. Good afternoon to respected chairman and all the dignitaries of this platform. My presentation is under the theme Recent Advances in Production and Protection of Crops under Open and Protected Condition. And the title of my presentation is Pruning in India's Hottest Chili Species, King Chili, Capsicum chinense, a promising technology for higher yield quality inside naturally ventilated polyhouse. So first, coming to the background information, I would like to draw a light on this species of chili. Actually, the northeastern region is recognized as a hotspot for chili diversity. There are many species of potential chili which are available in this region. Among these, one of India's hottest chili, that is King Chili, has been recognized in the Guinness Book of World Record also at one time as the most pungent chili species. And beside the pungency, this chili species also possesses a very pleasant and palatable aroma, and it also has a high vitamin C and capsaicin content. And this has made it an ideal chili for extraction of capsaicin. So in normal chili species, we find the capsaicin content like in a lesser percentage, but here in this particular species, the capsaicin content is reported to be up to 1 million for belly head unit, that is up to 6 to 8% more than the normal chili species. So, and this capsaicin is reported to have many pharmacological and medicinal properties also. So coming to the description and potentiality of this particular species, I would like to highlight that this chili species is a result of natural hybridization between two species, that is Capsicum chinense and Capsicum frutescens, but it is more closely related to Capsicum chinense. And as in comparison to the normal chili, which is annual in habit, this species of chili is a sort of semi-perennial type, and the growth habit is also robust, and so it makes it ideal for vertical utilization when we grow inside the protected structure. So coming to the importance and the potential of the grower, the premium price in market in during the main season, this chili fetches about rupees 500 per kg fresh fruit, and if it is produced in the off season using the protected technology, condition technology, it can, the price go up to 700 to 800 per kg fresh fruit. And when it is oven dry, it is costing up to 1,200 to 1,300 rupees per kg. So this highlight the importance and potential of this species. So now coming to the constraints, what constraints our farmers or the grower are facing in this particular species of chili? That is the, as I have already told, that it is a sort of semi-perennial type. But the main problem here is there is a reduction in fruit size and number of fruit after 5 to 6 months of fruiting and harvesting. And it is also sensitive to flood and waterlogging condition. So the cultivation in the open condition become a challenge for the grower and the farmers. So keeping in view these challenges, off-season cultivation and the scope of off-season cultivation for premium price, here comes into role the potentiality of protected cultivation. So there are early reports that when we grow the chili species such as the normal chili or the bell pepper and the Simla mirs in polyhouse, when they are given proper training and pruning, they give more yield and more early harvest also. So training and pruning can offer a better utilization of the vertical space inside a protected structure. It will also facilitate uniform light penetration, better crop management as the plant population is at the manageable number. Then utilization of space will be there. Plus there will be early harvest. Then high yield and quality of the fruit will also increase in terms of appearance and sizes. So keeping in view all this background information, the objective has been framed to evaluate the performance of this king chili species under different pruning system and to find out the ideal intensity of pruning to optimize the yield. So for this, in the evaluation trial, we have taken treatment which is comprising of 3 local advanced lines of king chili, that is ML3, ML5, and P2. And we are provided all these lines with a pruning intensity 3 in number, that is 2 stem, 4 stem, and the control is the unpruned. And the whole entire experiment was carried out under a low-cost polyhouse that is naturally ventilated, and the experiment was laid out in RVD with replication, 3 replication. So coming to the material and method, healthy and disease-free seedling were raised in the pro tray under protected structures, and they were
Dr.
transferred. Yes, sir.
Be brief, be brief. You
Okay,
are given only 8 minutes time. You have
Okay,
spent
sir. Okay, sir. Then they are transplanted in a spacing of 40 by 40 centimeter, and after 45 days of transplanting, these all the treatments were given, pruning at 2 stem, 4 stem at the interval of 10 to 15 days. And the statistical analysis was done as per Pense and Sukanbe. So coming to result and discussion, as per coming to the plant height, which is one of the important characters inside protected structure, it was found that the plant height was maximum, that is 183.33 centimeter in the 2-shoot system. Then coming to days to 50% flowering, this also 2-shoot system give the earliest flowering.
Are you through?
Yes, no,
Yeah.
sir. Coming to 50% harvesting, it was earliest in 2-shoot system, that is 80.50 days. And coming to number of fruits per plant, which is the most important contributing factor for high yield, 2-shoot system give the maximum number of the unpruned system give the maximum number of fruits per plant. Then in the fruit yield per plant, we observed that the maximum yield of 2.53 kg per plant was obtained in the 4-shoot system, followed by the 2-shoot system, and last is the unpruned system. Average fruit weight was highest in the 2-shoot system, followed by the 4-shoot system and the unpruned system. And coming to the capsaicin content, there was not much significant difference in all the pruning system. However, it was highest in 2-shoot system, that is 5.03%. So this is the picture showing the 4-stem pruning system in king chili inside naturally ventilated polyhouse. This is the vegetative growth after the pruning. And this picture also shows the vigorous fruiting of this chili species in the low-cost polyhouse. So coming to conclusion, we can conclude that there are many studies that has reported that pruning intensity in normal chili can give to early harvest and higher yield, but there was no such systematic research in this particular species of chili. So the present finding can be used for optimization of yield and earliness in king chili production under protected structure. That is all for today's presentation. Thank you so much. Thank you.
Thank you, Dr. Bilashini.
Thank you, sir.
Yeah, 2 questions.
Yes, sir.
Any 2 questions from the audience, please? If nobody is asking, I will ask you.
My question is, can you please explain the specific pruning technique which you have recommended for king chili plants in this polyhouse cultivation?
Okay, sir. Okay, madam. Actually, in the 4-stem system, madam, this actually chili species has the main 2 primary branches. After that primary branch, there are so many secondary and tertiary branches. So in the 4-shoot system, what we are doing, 2 primary branches are kept. And then from that 2 primary branches, we are pruning all the other branches and keeping only 2 from the side. Like for example, this is the 2 primary branches, and from these 2 primary branches, another 2 secondary branches were allowed. So this is the 4-stem pruning system, and in the 2-stem, we are keeping the 2 primary branches, and from that 2 primary branches, only 2 secondary branches were allowed to grow, madam.
Okay.
Yeah.
Dr. Swati, I have one question for you. Please, can you tell the specific time of pruning for particular crop?
Okay, sir. This is a very important question, sir. Actually, sir, this crops is semi-perennial, as I have told. So, during starting months, that is the first 6 months, it used to give a very good harvest. After 6 months of harvesting, the yield become reduced and size of the fruit also reduced. So, like for example, the growing season is starting from March till August. It is giving very nice harvest. So after August, like at the fall of September, we can give the pruning system. So by following this practice, in the next season, by early February, we are getting the very good vegetative growth and the early flowering also, and early harvest also we are getting. That means by fall of September, we have to cut off the branches at pruning.
Okay, good.
Yes, sir.
Dr. Bilashini?
Yes, sir.
You have presented this from which research station?
Sir, I have presented from Meghalaya headquarters, sir, ICR Research
Very nice.
Center.
I am
Yes.
very happy. I am very happy. I visited your center also.
Thank you, sir. Yes, sir. Sir, you are not audible, sir. Hello. Sir, you are not audible, sir.
Have you heard about Dr. Bijaya Devi?
Yes, sir. I know her, sir. She is my senior, sir, Dr. Bijaya Madam. Yes, sir. Yes, sir. Now she is in Tripura Center, sir.
Tripura Center. It's a very good work. While
Yes,
giving funding under NAT.
Sir, actually, your voice is interrupted, sir.
We have given a lot of funding for this program,
yes,
especially
sir.
when we were at ACRIPDA and ACRIPAM, and sometimes in NATP
yes,
and NAIP also.
Yes, sir. Yes, sir.
With one chili, people in the Northeast will pull on for 1 month. They will put a thread to the.
Yes, sir. I'm sorry, sir. Your voice is not audible to me, sir.
Really, this is a very good work, and you have to promote this. The government of
Okay,
India.
sir. Yes, sir.
इसके बाद के जो नेक्स्ट पार्टिसिपेंट्स हैं वो वन टू वन आते रहें, क्योंकि बार बार उनको बुलाना नहीं होगा, हमने लिस्ट पहले से ही दी है ग्रुप में, तो उस लिस्ट को अडेयर करें आप लोग और उस नंबर से आते चले जाएं.
Mr. Parul, are you there?
What is the price of
Okay,
this
sir.
particular in export orientation?
Madam, can you please repeat your question?
What is the potential of this particular king chili in the export?
Sir, madam, actually, export-wise, it has a very great potential. Recently, also, like our Meghalaya state government has exported to some like European and Arabian country also, madam. And as I have already highlighted in my slide, the oven dried fruit, when it is exported, it is facing up to rupees 1,300 per kg also, madam. And there is a huge demand, and for our institute has been approached by many like the private companies, even from Bangalore also we have sometime they have the request that if any
Thank you,
farmer
Dr. Bilas.
Yes, thank you so much, sir. Thank you, sir. Okay,
Really
thank you, sir.
appreciate your good work.
Okay, sir. Thank you so much, sir. Thank you, sir.
So Parul, are you there?
Yes, sir.
Oh, sorry, ma'am. We can start the session with you.
Next speaker, please. Next speaker.
Yes, sir. Yes, sir.
आप लोग वन टू वन अपनी पीपीटी रेडी रखिए आप.
Is my screen visible?
हाँ, स्क्रीन विजिबल है, आप कंटिन्यू करिए. आप इसको पीपीटी मोड पे भी डाल सकते हो, थोड़ा विजिबिलिटी बढ़ जाएगी इसकी.
सर मैंने किया है.
Yes.
ठीक है, आप कंटिन्यू करिए.
The PPT is moving.
Yeah, please go ahead.
Yes,
So-
it's visible.
Good afternoon, everyone. So, my name is Parul Singh, and I'm a PhD research scholar from the Department of Genetics and Plant Breeding from G.B. Pant University of Agriculture and Technology, Pantnagar. So, my topic is on influence of the nanosilica on the seed germination and seedling development in the soybean. So, first is this introduction. So, soybean is a very important miracle crop which contains the 40% protein and the 20% oil. It is grown globally. And India ranks 4th in the area and 5th in the production. Soybean is proven as poor storer, as the seeds of the soybean become unsuitable for the sowing just after 6 to 8 months of storage, and hence farmer cannot trust their own stored seeds for sowing.
Should be minimized, please. Go ahead with the important things so that your time will be apt, please.
Okay. That's what I'm talking about, ma'am. The seed is the simple seed treatment can bring the substantial improvement in the seed germination and the vigor of the soybean, because it is mechanically damaged and the
Ma'am,
field.
your slides are not moving.
Keep moving your slides.
Is it now?
Yes, please continue now.
So my objective, the experiment is, the first objective is to study the response of the nanosilica particle on the seed germination, and the second is to study the response on the seedling growth and the vigor. So I have taken 5 cultivars of the soybean, namely Bhat, Kalitur, PS26, PS1347, and PS1225. Bhat and the Kalitur are the land races, and the rest 3 are the promising varieties which are used as check in the field. Then these cultivars were treated with the different concentration of the nanosilica, that 800, 1000, 1500 ppm. Then observation was recorded by the towel paper method, and I have taken 3 replications and in one towel paper there were 100 seeds, and they were kept in the they were soaked for 4 hours, and then they were kept in the germinator at the temperature 20 to 30 degrees Celsius, and the relative humidity was maintained at 75 to 85%, and 2 countings were done. First counting was done at 5th day, and the second final counting was done at the 8th day. Then these are the characters which are recorded. The first one is a germination percentage. Second is the root length, shoot length, seedling length, fresh weight, dry weight, seedling vigor, and the seedling vigor index. The seedling vigor index is very important parameter for measuring the growth of the seed to find any that is measured by the 2 ways. The seedling vigor first is measured by the germination percentage and the seedling length. Seedling length is calculated by taking the average of the 10 seedlings of the root length and the shoot length of the average of the 10 seedlings. Then seedling vigor is calculated by the germination percentage into the dry weight of the 10 seedlings. Here is the table. Here we can see that the 1000 ppm has the highest effect on the seedling vigor, and it may be due to the because that nanosilica has a higher surface area, and it makes several holes in the seed coat that facilitate the quick absorption of the water, and due to this there is the breakdown of the stored food, resulting in the greater root length and the shoot length of the seed. But when we are going towards the higher dose of the nanosilica, the seed parameters, all the seed parameters are reduced. So so we can go through the optimum dose of the nanosilica. Then here you can these are the evidences of my experiment. In the PS26, this is a control, and this 80 and the 100 and the 150. Here is we can see the most of the 100 mg has the greater amount of seedling length. Then in the Kalitur also. Then in the PS 1347, in the Bharat, and in the PS 1225. In the end, I would like to conclude that our agriculture is threatened by the climate change and the depletion of the resources and the biodiversity. And so the nanotechnology has proven to increase the production of the crop as it works on the seed level, thereby ensuring the quality of food in a sustainable way. And the seed nanopriming is a very efficient process that can enhance the seed metabolism and the significant pathways affecting not only germination but also the seedling growth. And the nanopriming modulates biochemical pathways and balance between the reactive oxygen species and plant growth hormones, resulting in the promotion of the stress and disease-resistant outcome in the reduction of the pesticides and the fertilizer. Thank you.
Yeah, Dr. Ms. Parul. Hello, Ms. Parul.
Yes, sir.
Am I audible to you?
Yes, sir.
Basically, it's a good presentation. Have you done at the laboratory level, or you have done at the station?
No, sir.
I mean to say,
this was done in the genetics and plant breeding lab, sir.
Genetics and plant breeding lab. It is not done at Patharchatta.
No, sir.
Have you gone to Patharchatta?
Yes, sir. HRC, Horticulture Research Center.
Now that is at Patharchatta, no?
Yes, sir.
How is that?
So it has
plantations are in good state.
Yes, sir. And now, sir, but mine was in crop research center, no? CRC.
I understand. Yours is the laboratory experiment. I know that.
Yes, sir.
You have used silicon dioxide. I do know that.
Yes.
My general question is, are you aware that good amount of soybean is cultivated at these places?
Sorry, sir.
Do you know how much area is cultivated in Pannagar? These are the basic things before doing all these things. And you said biochemical pathways. What pathways are affected, or how do you account for the mode of action?
Sir, nanosilica, sir, act as a catalyst. As soon as there is it the seed coat touches the silica particles makes the holes in the seed coat, and the water enters in the seed, and all the biochemicals process starts that way, and the silica act as a catalyst to start the breakdown of the.
You are talking like a say lay person. You are going to make a good scientist. Make in India. We have to make you a good scientist. You have to give the scientific reasons. You have to explain. You have to explain what pathways.
What pathways? Sir, you're not audible. Hello, sir.
Hello, Miss Parul. We have one question.
Yes, sir.
Which state having a maximum area and production of soybean?
From Madhya Pradesh.
Okay. And production also?
Yes, sir. Production is from Madhya Pradesh.
Okay. Thank you. Thank you.
Next speaker.
Swati ma'am, are you there?
Each speaker kindly restrict yourself. Each speaker should restrict themselves to, each and everyone should restrict to 6 to 8 minutes, please. You give a brief intro.
Yes, sir, because the install it is taking a lot of time, and where we need to concentrate, there they are rushing.
Yeah, exactly. A brief, you have to give a passing remark about the introduction, then materials and methods, contemplate more on results and discussion. Okay? You finish your presentation in 5 to 6 minutes so that you can give 2 minutes margin for the questions and answer session. Thank you. Next speaker, please.
Thank you, sir. Swati ma'am, are you there?
Yeah.
Yes, sir.
Ma'am, your voice is breaking. Can you please share your screen and we can start the session?
Okay, okay.
अपना प्रेजेंटेशन शेयर करके आप प्रॉपर नेटवर्क जोन में पहुंच जाइए, क्योंकि अगर ब्रेक होता है ना सेशन तो थोड़ा सा उसका मैसेज बढ़िया नहीं जाता, और ये सेशन रिकॉर्ड हो रहा है पूरा. ये मैडम क्या पहली बार प्रेजेंट कर रही हैं आप? स्वाति मैडम, आवाज आ रही है मेरी आपको? आप रिस्पॉन्ड करिए, कोई वन वे फोरम थोड़ो ना है.
स्वाति मैम, आपका आवाज आ रहा है?
पूरे मोबाइल का आपने पूरी कुंडली दिखा दी, लेकिन प्रेजेंटेशन नहीं दिखाया आपने अभी तक।
तो वही गड़बड़ हो रहा है ना।
हाँ, डन स्पीकिंग। मैडम आप
Sir,
रहने दीजिए, नेक्स्ट को बुलाइए आप।
Kindly make the presentations from the PC or desktop so that it is easier for you and for us also.
या
Yeah,
एग्जैक्टली
yes,
मैडम,
sir.
बिल्कुल सही कह रहे हैं।
Yeah,
sir, is my slide visible?
yeah, you are visible and audible also.
Okay.
Go ahead, please.
Myself, good morning to everyone. Myself, Swati Sulakta Sarangi, Department of Vegetable Science from Odisha University of Agriculture and Technology. I am going to present assessment of the effect of organic nutrients and biofertilizers on soil properties and nutritional compositions of amaranthus. First of all, introduction. Amaranthus is fast-growing plant that is widely distributed throughout the world and belongs to the family of Amaranthaceae. It is widely cultivated green vegetable in summer and rainy season. Among the 70 species of Amaranthus, 17 are cultivated for edible leaves and 3 are cultivated as a food grain. Amaranthus leaves and stems are good economic sources of carotenoids, proteins, amino acid, dietary fiber, and minerals such as calcium, potassium. Then technical program. The experiment was conducted a randomized block design comprising of 11 treatments in 3 replication during the summer season. The plot size was 2 meter into 2 meter spacing 40 cm into 10 cm. Then fertilizer doses 60 is to 30 is to 20 kg NPK per hectare. Then treatment details. These are 11 treatments. Then some of pics of my experiment. I'm going to raise the effect of organic nutrients on nutritional compositions of amaranth. The treatment T4 recorded the highest 1.48% mean performance for crude fiber content, while lowest is treatment T11. Then vitamin C, the treatment T8 exhibited significantly the highest mean performance vitamin C content of the leaves, whereas treatment T11 is showed the lowest mean performance. Then calcium, the treatment T8 recorded significantly the highest mean performance for the calcium content with the value of 8.36, and it was at par with treatment T10. And the lowest the calcium content the leaves was observed.
Swati,
मैडम स्वाति, आपकी बिल्कुल आवाज नहीं आ रही है।
Swati, wind up. Wind up in 30 seconds.
सर वो हमें सेशन में है ही नहीं, वो वाइंड अप क्या करेंगी? नेक्स्ट को बुलाइए, रोहित जी आप नेक्स्ट को बुलाइए।
I think she got stuck up somewhere, sir, on the mobile.
हाँ
Yeah.
हाँ हाँ।
Next candidate, please.
नेक्स्ट कैंडिडेट प्लीज।
Sure, sir. So I'm just announcing the name of the participants. If anyone is online, please let us know. Okay. So Dr. Akansha Pandey, C. Bhanu Teja, Ashumi SR, Teshring Lahumu, Suma H, Rashmi, Pawan Saini, Imran Ahmad, E. Swati ma'am, the presentation is done, I think.
Swati? Okay. Next candidate, please.
So whosoever is ready can raise their hands. Participants, please raise your hands.
Participants, we are
चैट बॉक्स में जिन जिन कैंडिडेट्स का नाम लिखा है, अगर उनकी पीपीटी रेडी है तो आप प्रेजेंट करिए। हमारे चेयरमैन बैठे हैं, को चेयर्स बैठे हैं, हमारी फैकल्टीज बैठे हैं, आप लोग अपनी पीपीटी रेडी नहीं कर पा रहे।
We are closing the session by 1 p.m. So whosoever is ready, please come forward. Yeah. Go ahead.
मैडम हर्षदीप, एक बार ग्रुप में मैसेज कर दीजिए आप, और बड़ा क्लियर कट वे में करिए कि अगर जो पेमेंट नहीं करेगा हम उसका एक्नॉलेज नहीं करेंगे। हाँ, डॉक्टर टी मैडम, आइए आप अपनी पीपीटी को लेके मैम।
And one another user is Lenovo. Sir, please write your name.
जबकि मैंने एक स्ट्रिक्ट मैसेज डाला था कि रियलमी, ओप्पो, सैमसंग, आईफोन किसी से कोई ज्वाइन नहीं करेगा। हेलो, हाँ जी, आपकी आवाज आ रही है।
Haji, I am Dr. T.L. Bhutia. Can you please enable us so that we will be able to share my screen? I am not. Okay.
हाँ, आपको होस्ट हो गए, आप प्लीज अपनी।
Yes. Thank you. Okay. Is my slide visible?
Yeah. Please go ahead.
Yes,
Ms.
sir.
Lakshmi. Yeah.
A very good afternoon to the respected chairperson, co-chairperson, all the dignitaries and like students present in the today's international conference here. Myself, Dr. Chiringlam Bhutia. I'm like working as a scientist at IC Research Complex, NES Region Sikkim Center, Gangtok. So my topic of my presentation is ex situ conservation of rare and threatened orchid, that is Diplomeris hirsuta. So like the color of this orchid, it is like a snowy white. That's why like the name the common name of this orchid is also like a snow orchid, and it is a lithophytic orchid, which means like it grows on the rocks. So this Diplomeris, it has been derived from a Greek word that is divided stigma, and like hirsuta, it has been derived from the Latin word that is hairy. So it is found in India, northern Myanmar, southern China, northern Vietnam, northeast Thailand, Bhutan, and Nepal. In India, it is available in Sikkim, Arunachal Pradesh, West Bengal, and Western Himalayan region. So like at the right side of my slides, like it's a distribution of it's a map of distribution of this Diplomeris hirsuta like in the state of Sikkim and West Bengal. So in the Sikkim we have found at two places, that is like Pakcha, that is North Sikkim, and another is Assam Ringche, that is East Sikkim. Whereas in Darjeeling district, that's West Bengal, like it was recorded from Sibok Road. So this orchid species, it has large ovate oblong hairy leaves and usually produces single leaves only. So like occasionally only like we get two flowers per plant. So it usually grows like along the roadside, that is the wall along this roadside areas, in a cool and shady places on the moist rocks near running water streams, along with other vegetations like riverboats, ferns, and wild begonia. But unfortunately, like due to this like anthropogenic activities such as like habitat destruction, clearing of the land for widening of the roads, landslide, and scraping of these retaining walls for the beautification of the roads, this population of this orchid, it has become vulnerable, and like very much like vulnerable, and like till like it has like almost becoming now extinct. So there's no record of this conservation or successful rehabilitation of this species in ex situ environments. So like the rationale of the study is that like say ex situ conservation like is the practice of conserving a species outside of its natural habitat, and it is a valuable strategy especially for this orchid conservation. So this it can be implemented by cultivating these orchids, different orchids in specialized facilities like botanical gardens, greenhouses, and other controlled environments. So in case of this Diplomeris hirsuta, so there's a lack of information regarding its growth cycle, watering, and substrate requirements, which can be crucial for the successful ex situ conservation and its rehabilitation. So understanding the watering requirements of this endangered orchid species is very crucial for the successful conservation and rehabilitation. So to achieve this one, this Diplomaris hirsuta plants, they were grown in a controlled environment and subjected to different watering regimes. So such type of approach, it will help us understand the significance of providing optimal watering conditions for the species, ex situ conservation and rehabilitation efforts. By ensuring optimal watering condition, the survival and growth rate of this species, it can be improved, which will further increase the chances of the successful conservation and reintroduction again to its natural habitat. So therefore, this understanding the watering requirements of this endangered species, it's very much essential to conserve and protect them for the future generations. So looking into the importance of this orchid species and which have become very vulnerable and there's a fear for its extinction. So the following objectives, the objective it was kept for this present study, that is to conserve an endangered this orchid, the Diplomaris hirsuta, by creating a simulated natural habitat in the ex situ condition. So for the materials and method, for this study, Diplomaris hirsuta, it was collected from this natural habitat condition, and then natural that habitat condition was simulated or created, and wherein all these collective plants, they were grown on a bricks that was same naturally whatever the conditions of the nature, where the orchid was growing, the same it was replicated. It was the same habitat was replicated for its grown for the plant to grow. And this experiment, it was conducted in a semi-automatic glass house. And the treatments taken were watering in different watering rhythms, in February, and watering during March, and month of March, and watering in month of April. And there's various observations was also taken days to plant emergence, leaf length, and others. So what we obtained was that here this is the data of 2 years. Here we can see the leaf length and the leaf width, it was significant for all the treatments, and the maximum leaf length and the leaf width, it was recorded in the first year. It was recorded by the treatment number 1, that's watering during February month, and minimum was recorded by treatment number 3. And however, number of leaves, as I said, it is usually in the entire all the species, it is only one leaf, or maximum small, they have one small leaves, that is it is total of 2 leaves. So it was non-significant. Even for the days to flower withering also, it was non-significant. And then minimum days taken for this bud initiation, it was recorded in treatment number 3, that is during April watering. And maximum days was taken for treatment number 1. Similarly for the flower emergence also, treatment number 3, it recorded minimum days for flower emergence. So if you look into this the second year also, leaf length and leaf width, it was significant in all the treatments. And then here also, treatment number 1, it recorded the maximum leaf length and maximum leaf width, and minimum was recorded by the treatment number 3. Here also, treatment number 3 recorded minimum days for bud emergence. However, here, for the days for the flower emergence, minimum days were taken by treatment number 2. Here we can see, in the second year also, the leaf length and the leaf width was little bit smaller as compared to the initial year, the first year. So here it is little bit little more than the second year. And then here, actually, here what happens, this orchid species, once it flowers and the entire plant withers, so it goes into dormancy. It goes into dormancy, and it is only survived by the tubers. So once it gets favorable climate and water, it requires a continuous flow of water, then only it starts emerging in the next season. And then here, we can this what we observed was in this treatment number in the first year, this treatment number 1, and if all the plants, the treatment number 1, it emerged within May month, within the month of May, whereas treatment number 2 and 3,
Could you please wind up?
Pardon, sir?
Please wind up your results.
Yes, sir. Yes, sir. Yes, sir. And then here we said a number of this one, and even the number of this one, it was sorry, plant emergence was in the second year, it was earlier, within the month of May only, in all the treatments the plant was emerged. It is because due to this the climatic factors, because as I said, this this orchid, it is it requires warm and humid climate for the plant to grow. So here we can see in the as compared to 2018 and 2019, temperature was higher. That's why the plant emergence was also earlier in case in the second year. So like this is the like flowering in this ex situ condition, like successfully rehabilitated and conserved. So I would like to conclude from this like presentation and this experiment that like ex situ conservation efforts like were undertaken to preserve the species, and the study found that watering treatments played a crucial role in maintaining natural habitat conditions and promoting growth and survival of the plants. So this results showed that continuous water supply during the initial and the active growing stage led to the good vegetative growth and increased chances of survival. So this study provides us useful insights into the conservation of rare and endangered plant species and highlights the importance of ex situ conservation efforts in ensuring the survival of such type of species. Thank you, sir.
Very nice. It's a very good presentation. And we really appreciate the efforts of you, your team, and convey my, of course, DR Singh may not be there. Dr. DR Singh may not be there right now.
No, sir.
He's not there now?
So actually, I was working at NRCO before, like it's been 3 years now. I have been like transferred to NEH region, sir.
Yeah, very nice. Very, very nice. Sir. Have you heard about Dr. Foja Singh?
Pardon, sir?
Dr. Foja Singh has worked, my colleague at AHR Bangalore.
Yes, sir. Okay, sir.
A very good orchidarium has been developed. Orchids and
Yes, sir.
anthuriums.
Yes, sir. Yes, sir. In,
Yeah.
yes, sir. It is
Have you seen at AHR Bangalore?
like I have visited long time back on this, sir, not now. But I know, sir, like I have been like, I mean, I know that okay, good
There is a
quality.
very good orchidarium there. You please visit that. And
Yes.
Devinder Prakash, Dr. Foja Singh, they are the stalwarts who has initiated good work here in India.
Yes,
You please refer to their books, bulletins, and other things. Very nice. It's very nice. And when you have presented, you have to keep 2 things in mind, especially red plants, rare, endemic, and threatened plants.
Yes.
What is the potential? What is the status as on today? And way forward, where do you want to take it? Because good market is there, very good niche market is there, especially for the Northeast,
Yes,
with
sir.
regard to orchids and anthuriums.
Yes, sir.
You have to highlight that because your work will go unnoticed unless you give the economic importance of this. You continue this work and see that you you make use of this particular thing as one of the genetic material for future activities.
Yes, sir. Yes, sir. Yes, sir.
Keep going. All the best. Maybe one question from the audience, if anybody is having.
Dr. Saab, I have a question. Can you
Yeah, please.
tell me the challenges, the major challenges you encountered, and the challenges associated with ex situ conservation of orchids?
Yeah, very tremendous scope.
Ex situ actually, ma'am, this one like the challenges which I face, like actually this like the plant is already in like limited, no, ma'am, sir, and even in the like I mean, like in situ also, like it is like a population of these plants is very limited. So like we have to be very careful, like like taking out from that. And then that's why like our aim was to like like so that at least we can reintroduce the crop in this one, ma'am. Like that was the main.
Okay, thank you.
Good, very good. Next speaker, please. Next speaker. Kindly one of the co-hosts.
Yes, sir, I'm present, sir.
Share the screen, please.
I'm trying to share the screen. Sir, is it visible, my screen, sir?
Yes, ma'am.
Sir, am I audible, sir?
Yes, ma'am.
Yes, madam, perfectly audible.
Okay, thank you, sir. Good afternoon, everyone. I am Dr. Lama Laxmi, scientist senior scale in ICAR NEH Sikkim Center. And my today's presentation is on the topic genetic diversity studies of inbred lines developed from local land races of NEH region. So the northeastern parts of India fall under Eastern Himalayan region, and it is a biodiversity hotspot of many crops, including maize. So maize is an important traditional crop grown in the ecology of this region, and it is mainly cultivated for food, feed, and fodder. Maize in this region acquired different unique characters like colored grain having different ranges from purple, blue, white, cream, yellow, orange, red, and variegated. Some plants have multi-crop bearing traits having 3 to 6 crops per plant. mucus-producing roots having potential to fix atmospheric nitrogen, and vaccinous having high amylopectin content. In addition to the above traits, the local maize has high potential as fodder due to its vigorous growth and tillering habit, which makes them unique. The plant types of the local land races of this region ranges from wild primitive type to advanced land races of varieties maintained and selected by the farmers from the time immemorial.
Excuse me, ma'am. Ma'am, your slides are not moving.
It's not moving?
No.
Yeah, keep moving your slides, please.
Actually, I have moved from my side, sir. Is it moving now,
Yes,
sir?
yes, yes. Now it
It's
is
okay. It's okay now.
moving, ma'am.
Okay. The weather
Give less
condition, the
important.
weather condition of
Kindly
our
give less
northeastern region
importance for
are
your
very
introduction.
erratic. This drought and floods are very common phenomenon in this region. So accordingly, the local maize germplasm have evolved in balance with the prevalent agroecological conditions of the region, and therefore they are expected to possess better adaptation to regional stresses. So in order to tap the potentiality of such unexplored genetic resources, maize genetic resources, we have taken up this project to assess the genetic diversity using 30 early generation maize inbred developed from the local germplasm of Manipur and Mizoram using 52 microsatellite markers. So already I have told in the previous slides about the unique traits that is found in our local ranges, and here I have also depicted in the pictorial form. So coming to the materials and method, in my experiment, I have used 30 sets of indigenous local land races collected from different places of Manipur and Mizoram. And the genomic DNA isolation was done from the leaves using modified seed method. And for this experiment, we have used 52 SSM primers that are distributed across the whole genome. So for the agronomic performances evaluation, these 30 maize lines were grown with a spacing of 60 by 20 centimeter, following the standard package of practices for maize cultivation. For Sikkim, since it is totally organic state, so for the evaluation, I'm using only the organic manures and this biofertilizers only. So to avoid any contamination, we have carefully self the plant and harvested separately. And evaluation for various agronomic traits like plant height, ear height were also conducted. So for statistical analysis, we have used different programs like Servus, Genelex, Excel State, etc. So coming to the main part, that is a result and discussion. In the present study, all the 52 markers were successfully amplified, and a total of 189 alleles were generated. The allele size ranges from 50 base pair to 295 base pair, which reveal a high level of genetic diversity among the loci. The PIC value range from 0.7 to 0.76, demonstrating the high discriminatory power of these markers among the 30 lines. The heterozygosity percentage was also calculated, and it ranged from 0 to 27.45. Out of the 30 inbred lines, 30 lines show heterozygosity of less than 10%, and this means that around 43% of the lines have already attained more than 90% homozygosity, and the AMOVA revealed a higher molecular variance within individual of population than among individuals of population. So from the cluster diagram, you can see that the 30 maize genotype has been clustered into three different groups, A, B, and C. A having 2 subgroup, 2, and B having 3, and C having only 1. So most of the from this diagram, you can see that most of the inbreds derived from the land races of Mizoram fall in the same cluster, that is cluster V, suggesting the presence of high genetic similarity, and this may be due to the same selection history and the use of closely related populations to derive the inbred. So likewise also, we have also estimated the principal coordinated analysis, and this also revealed high genetic variation among these lines, with a total genetic variation of 18.05%. So from this, you can see that most of the inbreds derived derived from the population Manipur were found to be closely packed in the genotypic PCOA, while that from the Mizoram, they occupied the same axis in the same PCOA. So such pattern was also observed in the cluster diagram, which depicted that inbred derived from the same source population clustered themselves in the same group. So here I have given the description of the statistical data. I'm not going in detail because I have given the mean and maximum value, minimum, and the mean also. So we have also estimated the correlation coefficient. And from this table, you can see that the highest and positive significant correlation was observed between plant height and ear height. The association analysis was also done. And from this table, you can see that we have identified some major Q-tail for various traits like plant height, ear height, kernel roper, ear, etc. So this major Q-tail, this markers will be good. Will be useful for selecting lines having good agronomic characters. So coming to the most important parts of my findings, this is a very unique traits I have identified. So everyone knows about the symbiotic nitrogen fixation in leguminous crop like pulses and all, but atmospheric nitrogen fixation in non-leguminous crops like maize is still unknown to many researchers and farmers. So the mucus producing self-fertilizing maize was reported for the first time in the Sierra Mixte region of Mexico. And such plants, maize plants, have finger-like aerial roots that stick out of their stalks that secrete sugary like mucus, in which the bacteria and resides and that feed. So in return, the bacteria fixes nitrogen from the air
Dr.
and
Lama Lakshmi.
visible form. Yes, sir.
Dr. Lama Lakshmi.
Yes, sir. Yeah,
Wind
almost
up in
completed,
one
sir.
sentence. Wind up in one sentence.
Yeah, almost completed, sir. So many publications have done in regarding this mucus producing maize. So in our lines also, I have identified 3 lines. That is Mizoram 46B, Manipur 13, and MN this 3. This secretes mucus in the roots. So I have also published a paper regarding this, and we are in the verse of this registration of these 3 lines in the near future. So coming into conclusion, so I want to highlight that the lines developed from the local germplasm of maize germplasm may not be that much a good yielder, but it may harbor many useful traits which I have presented before. So these lines can be exploited as a donor in various maize improvement program, as well as this can be used as parents in hybrid development program. Moreover, these lines can be used in developing recombinant inbred lines. Thank you.
Thank you, Dr. Lama Lakshmi.
Yes, sir.
It's a very good work. I appreciate. First thing, I wanted to ask you a few questions.
Yes, sir. Please, sir.
Do you have any coordinated program?
Yes, sir. I have, sir.
Dr. Lama Lakshmi.
Yes, sir. Yes, sir. I have program, sir. I am dealing with rice, maize, buckwheat also, sir.
Not like that. I wanted you to connect with IISR, Indian Institute of Spices Research, Calicut.
Regarding
why don't you establish a coordination? Ask for it. You send your say theme paper and where exactly you wanted to connect to them. Otherwise, your work will go unnoticed.
Okay, sir. I will connect with them, sir.
You talk to your head of the institution.
Yes, sir.
You give your concept and send the concept note to IISR. If you want, I can also talk to the director, Indian Institute of Spices Research, Calicut. Okay?
Okay, sir. Okay, sir. Thank you. Okay,
all the best.
Okay,
Good luck.
sir.
Good work. Keep it up.
Okay, thank you, sir.
One question from audience.
Ma'am, could you please tell me in terms of practical application, what avenues for future research do you believe from your current work?
Oh, okay, madam. Already many inbred lines have been already developed that are having high yielding capacity. But this type of unique threads, sir, I'm telling about this stay-grain threads and all. No, stay-grain, it can be used for dual purpose, and that tillering maize that can be used as fodder, and that self-fertilizing maize already it is a very rare cases, ma'am. it is not found in all the germplasm. Only 3 lines have been identified. So this can be used in making this one. In one paper, I have read that up to this 70% of the nitrogen, that if the maize, particular maize is producing that mucus, then it can fix around 70% of the nitrogen. Its requirement, it can be fulfilled. So such type of this one unique traits can be incorporated in our crop improvement program. So apart from yield, then we can have this other unique traits like the dual purpose maize we can produce by incorporating this gene for this stay green trait like that, ma'am.
Okay,
So
okay,
it's a
ma'am.
very
Thank you.
much scope because our northeastern germplasm is not that much it is not that much explored, ma'am. So we need to explore them, ma'am, and we are making it in the inbred form. That is, we are fixing the gene, sir, ma'am. So
Right, right.
it has a very good scope, ma'am.
Ji, ma'am. Thank you so much.
Okay.
Next speaker, please.
Rashmi, are you there? Rashmi, ma'am? Ma'am, can you please unmute yourself?
Sir, am I audible?
Yes, ma'am. Yes, ma'am. Proceed.
And the slides are visible, sir?
Yes, ma'am.
Okay. So good morning to all. I am Rashmi from Punjab Agriculture University, Ludhiana, presenting research work on topics synergistic control of post-harvest fungal pathogens, evaluating combinations of Symbopogon flexuosus and Monarda cyclodora sensuae. So let's start with a brief introduction. Among the citrus growing countries, India holds 6th position worldwide, which includes the production of mandarins, grapefruit, lime, lemons, orange, tangerines, etc. So among all the citrus fruits, Kinnu is the most acceptable fruit due to its productivity, fruit quality, juice content, etc. But these fruits faces pre-harvest and post-harvest losses. The post-harvest losses can be due to improper handling, microbial attack, and packaging issues. So we are basically dealing with preventing the fruits from microbial attack. The most troublesome post-harvest fungal pathogens are Penicillium digitatum and Italicum, which are commonly known as green and blue mold respectively. So there are various synthetic chemical fungicides which have been used, but the practice of using these fungicides has that led to environmental problems and drug resistant strains. So therefore, employing natural products such as essential oil, bioactive components, and plant extracts, which belong to generally recognized as safe category, has been raised for controlling these fungal pathogens. So our previous studies reported good antimycotic potential of Symbopogon flexuosus, commonly known as lemongrass, and Monarda cyclodora, commonly known as lemon mint, essential oils individually against Penicillium digitatum. Therefore, the present study has been performed to investigate the combined possible interactive potential of both these plants against Penicillium species. So first of all, extraction and characterization of essential oil. Extraction was carried out by hydrodistillation, and the yield was found out by using this formula. And then the chemical composition of both essential oil were studied with the help of GCMS and GCFID, which revealed that citral was the predominant component found in Symbopogon essential oil, while thymol was the predominant component in Monarda cyclodora essential oil. Then coming to the antimycotic activity of individual essential oil on both fungal strains by using poison food technique, and their minimum inhibitory concentration were found out. Then the antimycotic activity in combination. So in case of combination, fractional inhibitory concentration index was calculated that involved the mix of one essential oil, half mix, 1/4 mix in combination with same proportion of other essential oil. So we basically took here 9 combinations and their FICI was calculated. If this value is less than 0.5, then it is considered as synergistic effect. Synergistic effect is something when the combined effect is greater than the individual effect. And if this value is greater than 0.5, this means additive effect. This means combined effect is similar. And if this value is greater than 1, it represents indifferent effect, which means partial synergy. And if this value is greater than 4, it is considered as antagonistic effect, which means that the combined effect is lower as compared to individual effect. So these are the results for antimycotic activity of individual essential oils. So complete inhibition of mycelial growth and conidial germination was displayed at simvastatin at 180 and 250 ppm, while Monada showed complete inhibition at 100 and 160 ppm respectively for Penicillium digitatum and Italicum. So the antimycotic activity of these essential oil could be due to the complex interaction of different classes of bioactive components present in these essential oils. So these are the parameters which are studied, that is number of colonies, mean spore forming region, radial mycelial growth, total colony diameter, and percentage inhibition. These graphical representation show the radial growth and morphological characteristics of Penicillium digitatum. Both the essential oils showed a concentration dependent decreased behavior for colony forming unit with increase in the radial diameter of the fungal colony. Similar trend was observed for Penicillium italicum. And then these are the results for antimycotic activity of essential oils when used in combination. So the results of combinatorial study also revealed concentration dependent interaction. Combination of both essential oil in different MIC blends displayed synergistic effect against Penicillium digitatum. Various combinations like MIC plus MIC, MIC plus half MIC plus MIC, half plus half MIC, 1/4 MIC plus MIC don't show any kind of growth, but these two combinations showed reduced growth, revealing their synergistic and additive effect when compared to the MIC values of both oils. But in case of Penicillium italicum, no growth was observed only in context of MIC plus MIC, while all the plates show growth in response to the MIC values. These are the FIC index values revealing these two combinations showing some kind of effect, that is synergistic and additive, while in case of Penicillium italicum, only this combination, while all other showing indifferent or antagonistic effects. Then the morphological characteristics of fungal hyphae was revealed by using the optical research microscopy. So distinct morphological variations were observed when comparing. Yes, sir. Okay. Distinct
there is a cross talking, please. No cross talking. No cross talking. Yeah, please go ahead.
Distinct morphological variations were observed when compared to control. This is control. The control represents clear cytoplasm and the conidial head are radiate and large. While in the treated fungal hyphae, it shows thinning and fragmentation of fungal hyphae where there is no conidial head. So these are the combinations which represents more retraction of cytoplasmic content, and these are the concentration values for essential oils. Similarly seen in case of Penicillium italicum. So these are the in vivo study. This is just an initial scrutiny which we did. We placed inoculated fruit in the center, and all the fruits which were placed in the periphery are bruised in the ring on the opposite faces, and then we apply our combinations as well as the MIC concentrations of essential oil. So we found and evaluated in further growth of the pathogen, and we found that at the 6th day there was growth on the MIC values of both essential oil, but there is no growth on the combination on the kinu treated with the combination concentrations. So this is generally initial scrutiny. Our in vivo long-term experiment in the cold store conditions is in progress. So the conclusion, synergistic effect of essential oil has been observed against Penicillium digitatum, and the combinations of both essential oil, these two combinations which show reduced growth as well as the other combinations which show no growth could be used as eco-friendly alternate to enhance the shelf life of tea. So thank you.
Hello.
Hello, Hanji sir.
So madam, I have one question for you. What will be the probable cost per kilogram of quinoa if you are applying it on quinoa? And what will be the probable cost, and what do you think, what will be its shelf life?
Sir, as I already told that if we are using simple essential oils,
Yep.
then the shelf life is around, if we are like in vitro experiment में sir हमने देखा कि 14 days तक we can prevent the quinoa. But if
Okay.
we are using combinations, then we can prevent the fungal growth till 20 days, like
हाँ,
post 18 days,
आप
18 or
इसको
post.
आप इसको कंट्रोल कंडीशन में रख रहे हो?
Yes sir, in vitro
हाँ,
experiment in like fungus की actual जिस
ओके,
temperature पे वो grow करता है, और
हाँ जी,
cold condition पे हम 60 days तक देखते हैं कि कितनी
ओके,
fungal growth हुई है कि नहीं.
ओके। और इसकी प्रोबेबल कॉस्ट क्या रहेगी पर केजी?
सर जैसे essential oils तो मतलब 300 to 500 rupees पर 10 to 20 ml अवेलेबल हो जाते हैं.
नहीं,
और
तो
हमें
आप इसकी कोटिंग भी करोगे तो अल्टीमेटली आपको पर केजी कितना पड़ने वाला है? इसकी मार्केट अवेलेबिलिटी तो, या फिर मार्केट, इस पे मार्केट में जाएगा कि नहीं, तो इस चीज़ के ऊपर डिपेंड करेगा ना कि आप पर किलोग्राम आपके चार्जेस कितने आ रहे हैं, आपका ब्रेक इवन निकल रहा है कि नहीं, तो उसका क्या कुछ रहेगा?
सर जैसे 300 to 500 रुपीस अगर हम ऑयल का ले रहे हैं तो उससे
हाँ जी,
100 kg किन्नू कवर हो सकता है आराम से.
100 केजी।
हाँ जी, इफ वी आर यूजिंग वाटर और इफ
ओके,
वी आर यूजिंग वैक्स सिमिलर.
ओके, ठीक है, थैंक यू।
Thank you very much.
Okay, thank you sir.
Next speaker, please.
Yes, Mr. Baljeet, are you there?
Sir, my PPT is visible?
Yes, yes, sir.
Good morning to everyone. My name is Baljeet Singh and I am a PhD scholar from Soil and Water Engineering Department. And my presentation topic is role of lateral impact strategies on discharge variation of sprinklers for smooth pipes. As we know that sprinkler irrigation is a method of applying water in the form of spray or like rainfall. And sprinkler system, it is most widely irrigation method which depend on the lateral design strategy to achieve uniform water distribution. And in this system, the pipe size mainly are selected to minimize the pressure variations along the lateral, because due to friction losses and due to also elevation changes. And generally, it is recommended that the total pressure variation in the lateral practicable should not be more than 20% of the higher pressure. And so it is necessary to compute the friction loss in the lateral line where flow decreases along
Baljeet,
its.
your slides are stagnant, they are not changing.
Sir.
Not yet.
No,
No,
sir.
no, it's okay. Please
Okay.
continue. Yep.
So the Hazen-Williams and Darcy equation are both used to calculate head loss due to friction in fluid flow. Mainly the Hazen-Williams equation is commonly used for the water flow through pipes. And then while the Darcy-Weber equation is more versatile and accurate method for calculation of head loss due to friction in fluid flow as compared to the Hazen-Williamson equation. So, and the another factor is design pressure also play an important role in sprinkler performance. And then by consideration, the design pressure at different locations, such as at the first sprinkler, at the middle, and at the last sprinklers. So these approaches help to avoid the different issues such as inadequate water coverage, uneven irrigation, and potential damage to crops due to insufficient or excessive water pressure at different points along the lateral line. So the design pressure for laterals, first is selection of different lateral length and diameter, then selection of different sprinkler discharge and spacing, and then the calculation of head loss using by different equation, and then the calculation of available head at the different sprinklers, and then calculation of flow rate, and at the last the calculation discharge variation of sprinklers. And then the head loss is calculated by using two different equations in this study. The first equation is used for Hazen-Williamson equation, and the second equation is used Darcy-Weisel equation. And the friction factor for the different equation is y calculated by the different scientists. First one is the Blaise equation. If our real number lies between the 3000 to the 10 raised to power, then the friction factor calculated by Blaise equation. If it is lies between 10 raised to 5 to 10 raised to power 7, then it is calculated by the Waiter and Keller equation. And so the Reynolds number is calculated by the Roy equation. And after that, the sprinkler discharge is calculated by the equation first. In this case, is the discharge coefficient for sprinkler nozzles, and then the discharge variation is also calculated by the equation second. And then the table 1 shows the effect of different lateral length on discharge variation of sprinkler. In this equation, we kept the diameter of lateral is 50 mm, and sprinkler discharge kept as constant 0.78 lps for different lateral length, and the operating heat is kept at 30 meter, and the sprinkler spacing is taken as 10 meter. And in this for this calculation, the design pressure taken at the last sprinklers. In this table, we saw that if our length of lateral is increases, the discharge variation is also increases. And then table second show the effect of different lateral diameter on discharge variation of sprinklers. In this also we can say that the lateral dia increases, the discharge variation are also decreases. But at 100 and 125 mm, we can see that the discharge variation is same for both the equation as in Williamson and Darcy equation, 1.52 to 1.50. And table 3 show the effect of different sprinkler spacing on discharge variation. As we can see that if the sprinkler spacing is increasing, our discharge variation is also decreases. And then table 4 show the effect of different combination of different lateral length for sprinkler spacing, and also the location of design pressure at different locations. At last location, then at middle, and then at first sprinkler. In this table, we can see that the maximum discharge variation was found if we take our first sprinkler if we take our design pressure at first sprinkler, and it is found minimum if we take the design pressure at the last sprinklers. And then conclusions. We can conclude that lateral length has a significant impact on discharge variation, with a longer length leading to higher discharge variation values, and the discharge variation of sprinkler decreases as the lateral diameters increases. And then the discharge variation values are found to be higher when the design pressure is taken at first sprinkler, and lower when the design pressure is taken at the last sprinklers or middle sprinklers. Thank you.
Thank you very much, Dr. Baljit Singh.
Okay,
Thank you very much. It's a good presentation from Soil Water Conservation Engineering. Could you please tell, for 2 hectares of land, 2 hectares, I use the word 2 hectare in a square form, 2 hectares in a rectangular form of different spaces. Could you please give a model?
Yes, sir. If it depends on mainly on the different crop types.
Okay.
The maximum we can kept in this sprinkler.
Okay, for 2 hectares, what should be the pump required for drawing water, especially from a bore well, not canal water?
The exact ध्यान is 7 HP approx.
7 HP.
Exact
if it is 7 HP, if it is online dripper, what should be the diameter for a quinoa crop or sweet orange crop or mango crop, whatever fruit crop it is, at a spacing of 5 meter by 5 meter and 10 meter by 10 meter. And they are adult plants of 10 years age. Do you have any ready reckoner? So these are the things which it will go as a recommendation.
Okay.
Simply doing something, whatever is there in our field, will not help other people. Because when you say that you are from Hisar, when you are from Haryana Agricultural University, people expect that there should be a recommendation from your research. Is it not?
Yeah. Yes, sir.
Yeah. Okay. If it is 7.5 HP, how much pressure should be given online with 8 LPH capacity and 16 LPH capacity? Have you devised that? Dr. Baljeet Singh?
Hello, sir.
Yeah, आप सुन रहे हैं ना? मुझे सुन रहे हैं आप?
हाँ, yes, sir.
Yeah. Could you please give? Or else you prepare. Whatever you give, whatever experiment you do, it should be applicable on a wider scale. Think that way and give the conclusions. Then they go as recommendations. When you are presenting in international conference, these are expected of from any research paper presented here. Is it okay, Dr. Sir?
Okay, sir.
Right. Next speaker, please.
Dr. Suma, are you there? Dr. Suma?
The next speaker could be given chance, sir, if they are not there. Yeah, she started sharing the screen.
I think network is not good, ma'am. Okay, we can proceed with our next participant, Dr. Chetan K. Mr. Chetan, are you there, sir?
Yes, sir. Yes.
Okay, I'll make you co-host. We can start with you.
Sure, sir.
Okay, thank you.
Is my screen visible, sir?
Yes, 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 yes.
Good afternoon, everyone. I am Chetan from Division of Plant Pathology, IARI, New Delhi. Today, I am going to present about my part of my research work, that is effect of set bacterialization on sugarcane seedlings under greenhouse condition. This is a part of my MSc research work. And when come to sugarcane, it is a important crop, commercial crop, cash crop. It is a main source of sugar in the country. And even with that much importance, there is a major hindrance from the biotic stresses that will decrease the sugarcane production. Among the major biotic stresses, wilt in sugarcane is caused by Fusarium saccharine contributes more yield loss in case of sugarcane. The symptoms includes the turning of leaves turning yellow and shrinkage of internodal tissues, and finally drying and death of the sugarcane that stalk, which is the economical part of this crop. When come to its management, even though we are having many chemical fungicides in order to control this, due to the strong rind of this sugarcane sets, the penetration power of chemicals was decreasing, and their environmental effects made it less user. Among the viable methods, this plant growth promoting rhizobacteria will help us to control this sugarcane. In this regard, we have planned our experiment in order to isolate, evaluate, and in order and characterize those rhizobacteria that can be used to control this disease in sugarcane crop. Plant growth promoting rhizobacteria is an indigenous rhizoplane bacteria that can have two different kind of effects on this plant growth. One is direct ways like biocontrol, antibiosis, lytic enzyme production, and induced systemic resistance. And in also in indirect ways by strengthening the plant by providing nutrients like phosphorus solubilization, nitrogen fixation, siderophore production in this kind. When come to the design of experiment, first we have taken the rhizoplane soil sample. From that, we have done serial dilution in order to isolate the bacillus and pseudomonas on pseudomonas and bacillus specific agars using the selective supplements. From there, we have obtained the pure culture of these bacterial isolates. Then we have conducted dual culture studies in order to check their antagonistic efficacy against this fusarium pathogen. And then we have for under greenhouse studies, we have treated the single noded sets with the bacterial cell suspension, and we have conducted experiment on portraits, and we have calculated the seedling vigor index at the seedling stage. When it comes to results and discussion, this set bacterialization among the bacillus isolates, we have seen a negligible to higher increase in germination. Among the 20 bacillus isolates, the B11 isolate have shown extraordinary performance in case of increasing the germination percentage up to 60%. It has increased than the control. Next, among the Pseudomonas isolates, there we were isolated both 20 Bacillus isolates and 20 Pseudomonas isolates. Among the Pseudomonas isolates, we have seen negligible to higher increase in germination. And among this Pseudomonas isolates, this P20 isolate is has performed very well. When it comes to the bacillus isolates, in case of shoot length, we have observed very negligible or very low increase in shoot length when compared to control. And among them, B11, as we seen in case of germination percentage, the same isolate B11 has performed well. But very interestingly, in our research, these are some of the isolates have shown negative impact on the shoot length that we are going to discuss in later part. And in the same, and in among Pseudomonas isolates also, we have seen negligible to very low increase in shoot length. And here also we have seen negative impact of some isolates on shoot length. This is because of that these microorganisms not only promote or not only provide nutrients to the plants, but sometimes they may compete with the plants for that nutrients also. And the thing is, these microorganisms create a micro environment around the rhizosphere, so that it may change in the pH and other physical and chemical properties of the soil, so that it will hinder the nutrient uptake by the plants, that may affect in this decreased in shoot length. When comes to seedling vigor index, among the bacillus isolates, we have seen a very low to very high increase in seedling vigor. Among them, bacillus isolate, the same B11 isolates has performed very well. And we have seen, we can see many more isolates having more than 50% increase in seedling vigor index. They can be used in a consortium in order to increase the growth in case of sugarcane. And when it comes to the Pseudomonas isolates, here also it is a low to higher increase in seedling vigor index. The thing is, here the isolate SRP17 has performed very well. And here I am going to concluding by saying that the isolates SRP2, 4, 20 and the 3 Pseudomonas isolate, SRP 3, 19, and 20, they have performed very well in this seedling vigor index, which are already having antagonistic efficacy against fusarium wilt of sugarcane and also red rot of sugarcane. Those were our findings from our previous study. In this study, we have evaluated these isolates for their plant growth promoting activity, and we are concluding that these isolates are having both antagonistic efficacy and also plant promoting efficacy, acting in both direct and indirect way of biological control. And these isolates were identified as Bacillus inquisitorum, Bacillus valismotis, and Pseudomonas chlororaphis and Pseudomonas aerogenesa that were molecularly characterized in our further studies. And among future prospect, these isolates has to be tested under field condition, and also the experiments on formulation is under study for these isolates. Thank you.
Thank you, Chetan.
Yes, sir.
You have very well adhered to the timing. Very nice. What is your recommendation?
Sir, the
As a researcher.
recommendation. Yes, sir.
2 sentences.
Yes, sir. The recommendation is we have found out one Bacillus isolate and one Pseudomonas isolate, that is Bacillus valismotis and Pseudomonas chlororaphis. The consortium of these 2 bacteria can be a better management of these soil-borne diseases in case of sugarcane, sir. That will help in both this controlling disease controlling activity and also growth promoting activity. So we no need to take different measures for different pathogen. As in a collective form, we can work through, sir.
Okay. Don't you think there have been very good recommendations in the past also? They should be done away with?
Yes, sir. There are
different consortia were.
Yes, sir. Actually, in case
different
of
consortia were promulgated in the past.
Yes, sir.
Especially from your own lab at IRI, microbiology.
Yes.
Yeah.
Yes, sir. But actually, now I am pursuing my PhD degree in IRI, sir. But I worked in RARS Research Station, Anakapalli, Visakhapatnam. The thing is, there they have developed many trichoderma isolates and trichoderma consortium, fungal consortium against these diseases. But the bacterial antagonist, there were many less work in case of sugarcane. That's why we worked on this, sir.
Did you work in Meenakshi Grover's laboratory?
No, sir. Regional Agriculture Research Station. No, sir. Anakapalli, Visakhapatnam, sir.
So you didn't work at IRI main campus?
No, sir.
Okay. It's a good presentation. One question, quickly, if anybody is having.
Yes, sir.
Only one question.
This is from Dr. Bhandarekha. Dr. Bhandarekha, yes. Sir was asking.
Yeah, please go ahead.
There could have been some evaluation with the already existing effective ones, because all of a sudden we cannot exclude the older ones which were performing well and say that this new strain is most effective for this particular control of the diseases.
Yeah.
Pardon, ma'am?
So you were recommending that these particular two strains are more effective, isn't it, for the wilt control?
Yes, ma'am. Yes, ma'am.
But previously also there are several things which were recommended. I hope that these studies should include the older ones which were performing well, so that we can compare along with the already existing recommended things.
Yes,
Not
ma'am.
just new things which have come across.
Yes, ma'am. We are in that process, ma'am. Actually, those already existing isolates were already in field condition, so that now our new isolates are now going for field condition. So we are going to evaluate in future under field condition how it works when compared to the previous isolates. In future, we are going to do that.
You are going to evaluate in connection with other consortia. Okay, thank you very much.
Yes,
Next
sir,
speaker, please.
Thank you.
Sir, is my screen is visible? Sir, is my screen is visible?
Yeah, visible,
Yes,
madam.
madam.
It is visible.
Okay. Good afternoon, one and all. My name is Dr. Suma. Today, I am here to present on the effect of different combinations of raw banana and whey protein on its physical attributes to make an instant drink powder. First of all, I have taken raw banana, whey protein, and drumstick leaf because these three ingredients are very nutritious and they are easily available. First of all, banana is a rich source of potassium and it has many health benefits such as it is rich in vitamin A, vitamin B2, some minor nutrients such as calcium, magnesium, phosphorus, and it also helps in the constipation, diarrhea, and dysentery. And it also helps in the to overcome the malnutrition. Next comes the drumstick leaf. Drumstick leaf contains highest amount of calcium and iron. So we can, as it is a storehouse of many important nutrients. Sir, am I audible?
Yes,
You are
madam.
audible, but the slides are not moving.
Not visible, ma'am.
Visible, ma'am.
Okay.
But
Next
please
one.
move the slides also.
Okay, ma'am. Now, the next ingredient which I took was whey protein. Whey protein is highly rich in protein. It's a small fraction of individual protein content. It is separated out from the cheese making. We will separate out from the casein while making the cheese. So it is highly rich in protein. So here are the some of the treatment details. From T1 to T9, I have taken T1 is devoid of raw banana and drumstick leaf powder, but it contains 50% of the whey protein concentrate. From T2 to T9, it contains same amount of raw banana, but there is a difference in the content of drumstick leaf powder and the whey protein concentrate. But in all the treatments, carboxymethyl cellulose and sugar powder is common. Here, I took raw bananas, I peeled the raw bananas and I kept the raw bananas in freeze dryer to make the powder out of the raw banana slices. The next one is drumstick leaf powder. I took the drumstick leaves, I washed them, I took the shade drying of the drumstick leaves and I went for the freeze drying of the drumstick leaf to make the powder out of the drumstick leaf powder. Here, these are the ingredients which are used in the to make the instant drink powder. One is whey protein, one is drumstick leaf powder, and another one is raw banana powder, along with CMC, carboxymethyl cellulose, and powder sugar. Here, these are the different treatment combinations, and this is the different treatment combinations when we mix the instant drink powder in water. Here the table 1 represents the bulk density and the tap density and also the flowability of the powder. From in the bulk density and tap density will tell us regarding the heaviness of a material. From treatment T1 to T9 we can see that T1 to T8 all are on par with each other, but T9 is significantly different from T1 to T8. This might be due to the incorporation of whey protein concentrate. And when it comes to the flowability is judged upon the car index. Car index is a index which we will see different flowability of the powder according to the index given. Less than 15 means very good, 15 to 20 good, 20 to 35 fair, 35 to 45 bad, and more than 45 is very bad. So in case of flowability, the T1 has the bias very bad flowability because of the high content of whey protein. Whereas from T2 to T9, we can see that the flowability is decreasing, means it has a fair flowability. This is because of the incorporation of drumstick leaf powder. Here, this is the graph which tells about regarding the car index and the Hausner ratio, which we'll explain in the next slide. Here, this Hausner ratio is done in order to check the cohesiveness of the powder. Cohesiveness means the how much the powder is cohesive, how the particles are cohesive to each other. Here you can see in treatment T1, it is having the highest cohesiveness. This is due to the incorporation of whey protein concentrate. Whereas other treatments are having the intermediate and the high cohesiveness. This is due to the increase in the content of whey protein concentrate. Where it comes to the angle of repose is the parameter where it will tell about the pileness of the materials in the instant drink powder. How they will get piled means when we take the powder, they are highly piled or less piled. and it also determines the flowability. Here in T1, again, we can see the poor flowability because of the more content of whey protein concentrate. Whereas from T2 to T9, we can see the excellent flowability because of the incorporation of drumstick leaf powder. Here in table 3, this is regarding the color values of the instant drink powder. Always people will prefer that they should like the appearance of the instant drink powder. So therefore the color values are very important. Here you can see the L star will indicate the L0 will indicate the white color and L100 will indicate the black color. Here you can see the T1 is having the highest L star value because of the higher amount of whey protein content. Whey protein is white in color. That's why it is having the highest L star value. Whereas from T2 to T9, you can see it is getting darker because of the incorporation of drumstick leaf powder. Whereas A star will indicate the green to red. Means A negative is green. That's why we can see from T4 to T9, it is negative values. Negative values indicating that it contains the drumstick leaf powder. Whereas B star will indicate the yellowness to blueness. Here you can see the values are increasing from T2 to T9 because of the high content of whey protein is getting increased in the drumstick leaf powder combination. And based on this, I would like to conclude that T4 is showing from T2 to T9, it is showing the fair flowability, whereas T1 is showing the very bad flowability, very bad cohesiveness because of the high whey protein content. Whereas T1, T3, T6, T7, T8, and T9 are showing the high cohesiveness. Whereas T2, T4, T5 have intermediate cohesiveness. Thank you. Thank you, one and all. Hello.
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 Madam Bhanu Rekha, please.
हाँ sir, I concluded sir.
हेलो मैम, आपने ड्रम स्टिक लीव्स को पहले शेड ड्राई किया फिर फ्रीज ड्राई किया, इसका कोई रीजन था?
Sir, because shade dry sir, जब हम लोग when we wash the drumstick leaves, they have some kind of water in it.
Okay, just remove the moisture, initial moisture where present on its surface, huh?
हाँ, just a light, just not completely removal sir, just a shade dry for like 3 hours.
Okay.
So it will remove the moisture, and when we go for freeze drying, it will be better drying conditions we can see.
And there was a slightly mistype on your presentation. L0 indicates black color and L100 indicates white color.
Yes,
in
sir.
the diagram, you by mistakenly, I think you mentioned that L0 is white.
Sir, L star will indicate positive will indicate.
Yes, I'm getting it. It changes from black to white. L is equal to 0 is black and L
Yes, sir.
is white.
Yes, sir.
Mentioned it wrongly.
Okay, sir. Yes, sir. Because of the increase in the content of drumstick leaf powder, it will show little bit dark in color, sir.
Madam, one doubt I have.
Yes, ma'am.
Actually, nowadays they are using a lot of this moringa dried things and all.
Yes, ma'am.
So, what I meant to say is, this color, what you are saying, that is also one of the drawback. What we are seeing, that it is giving a very dark color to the end product.
Yes,
so
ma'am.
there
Yes,
is
ma'am.
any remedies which we could reduce that color, because unless and until it gives a attractive color, the product
Yes,
will
ma'am.
not have good demand in the
Yes,
market.
ma'am. Sure. Yes, ma'am. Yes, ma'am.
What is
So,
that which we could follow for it?
To decrease the color, ma'am.
Yeah, to decrease the color and to have an attractive color.
Ma'am, to have an attractive color, like ma'am, if we balance it with whey protein concentrate, ma'am, then we can balance the color, ma'am. Otherwise, like it's very difficult to decrease the color of drumstick leaf powder, ma'am.
What was that with respect to your product color? Was there any discoloration or tinge of the color which you have done with this particular product?
No, ma'am. I didn't get your question, ma'am.
So you had also a blend of this moringa leaf powder, isn't
Yes,
it?
ma'am. Yes, ma'am. Blend.
So, the range of the color which you could see, was there
Yes,
any
ma'am.
difference with the increase in the proportion and so on?
Yes, ma'am. Yes, ma'am. When I was increasing the content of drumstick leaf powder in the different combinations, ma'am, it was showing more green color, ma'am. Means negative values.
So with respect to color also, whatever one you are referring is also okay with the color also?
Ma'am, when I went for the sensory evaluation through the some panels, they will like okay with the color, ma'am, because as the chlorophyll will dominate, ma'am. So the sensory panel was okay with the color, ma'am.
Madam, your end results were based on optimization or standardization. What have you done? How you
Sir,
recommended that?
your voice is very low, sir. Like volume
Okay,
is
okay,
very.
okay, okay, okay. Now it is clear.
Now it's clear, sir.
I'm saying that that the last combination you have recommended, is
Yes,
it
sir.
based on standardization or optimization? What have you done?
Sir,
Because
not on standardization, sir. Based on the
okay,
parameters which I have taken, like cohesiveness, bulk density, tap density, based on this,
uh,
I'm telling
I I think if you do optimization, then you'll get the exact proportions that you can mix up. Optimization will be a better option.
yes, sir. That will
Okay.
be a yes, sure,
Yes.
sir.
Yeah. Thank you.
Sure, sir. Yes.
Ma'am, can you please provide some other examples of innovative formulations featuring the ingredients which you have taken, like raw banana and whey protein, or instant powder drinks?
Other combinations.
Yeah.
Other combinations, ma'am. We can have a blend of kokum, ma'am, because kokum has a rich color also and has a flavor also, ma'am. So we can have a blend of kokum. Kokum is rich in anthocyanins also. It is good for health also. So a blend of kokum will be a better option, ma'am.
Are there any other products like which the ingredients which you have taken in your research?
Oh, okay. Along with my ingredients, ma'am.
Uh,
Other like, ma'am, they have instant millet mix, ma'am. They have taken the raw banana powder along with the drumstick leaf powder, along with millet powder they have taken, ma'am. Millets powder like ragi powder they have taken combination.
Right, right. Mhm.
Jowar powder and millet mix they have taken, ma'am, along with this.
Mhm.
Again, we can add through millets, we can add another nutrition level to the instant drink powder.
Right, right.
Yes.
ठीक है नेक्स्ट पार्टिसिपेंट्स को बुलाइए
Ashwini, ma'am, are you there?
Yes, sir.
Okay, we can start with you. Dr. Ashwini.
Yes, sir. Sir, is my slide visible? Is it
Yes,
moving?
ma'am. Yes, ma'am.
A very good afternoon to one and all present. Thank you for giving me the opportunity to present my oral talk on shelf life extension of Prunus nepalensis by post-harvest application of surface coatings. Myself, Dr. S. Ruth Asumi, I'm scientist horticulture subject in ICRRC for NEH region from Nagaland Center. Prunus nepalensis it is locally called Sohyong in Meghalaya. This experiment was conducted when I was posted in Meghalaya last year. It's considered as an important underutilized crop indigenous to Northeast region. And Sohyong is a potential fruit due to its unique quality, attractive color, pleasing flavor, and also it is rich in anthocyanins, vitamins, minerals. Particularly anthocyanin is stable when we have attempted to make value-added products like beverages and dehydrated bars. This fruit contributes significantly in the traditional food basket and is believed to be rich in bioactive properties, antioxidants also. And we have also studied that it is present in antiproliferative activity also. However, this potential underutilized fruit is challenged by a very short shelf life. And then we have attempted this study so that we will be able to extend the shelf life of this harvested fruit. And the work on post-harvest shelf life extension of this underutilized fruit is also very limited. So the present experiment was undertaken to study the influence of polysaccharide-based edible coatings, that is guar gum, carboxymethyl cellulose, sodium alginate, and tyrosine on the storability of Prunus nepalensis in ambient conditions. So there were 4 treatments. T1 was 1% carboxymethyl cellulose, treatment 2 was 2% sodium alginate, treatment 3 was 1% tyrosine, and treatment 4 was guar gum. And it was given the chlorine-based wash before the treatments, and the surfactants used were glycerol 2020 and glycerol acetic acid. And the post-harvest study was done at 2 days intervals, and the parameters considered were PLW, physiological loss in weight, And texture and firmness were also tested with a texture analyzer, the total soluble solids and acidity, and also the pectin methyl esterase activity and the total anthocyanins. So the methodology was we have selected the mature uniform ripened fruits of Prunus nepalensis, and then we have selected uniform sizes, and we have given the treatments, the 4 treatments, and also the control. And after the treatments, the fruits were air dried, and it was stored at room temperature, ranging from 21 to 24 degrees Celsius, with 80 to 90% relative humidity, and the fruits were stored for up to 8 days. So the study at 2 days interval up to 8 days showed that the physiological loss in weight of Prunus nepalensis during the ambient storage was best performed with guargum 1% treatment, and the control showed the highest PLW. And when the PLW reached 12%, we observed that the fruits became unacceptable for commercial sale. And with respect to texture and the firmness of Prunus nepalensis during storage in ambient conditions, here also the guargum 1% fruits performed best, and least firm was with the control fruits. So this is a pictorial presentation of Prunus nepalensis from 0 to 8 days, and the best performing shelf life after 8 days was recorded in Prunus nepalensis with guargum 1%. At room temperature storage. And also with respect to the effect of edible coatings on the pectin methyl esterase, a very important cell wall enzyme, it showed that the minimum activity of PME in Prunus nepalensis was associated with the guargum 1%, followed by chitosan 1%. So with this small experiment, we can come to the conclusion that the coatings of guargum 1% and chitosan 1% significantly delays the weight loss and maintain the texture. The use of this edible polysaccharide-based coatings maintained lower activities of pectin methyl esterase, a cell degrading enzyme. And results indicated that this use of edible coatings is a promising strategy for improving the post-harvest quality and extending the shelf life of fresh Prunus nepalensis fruits in ambient conditions. Thank you. This is all from my presentation.
Hello.
Yes, sir.
Madam, you have only considered physiological loss of weight as the indicator of shelf life?
And also the texture and the enzyme activity, as we are restricted with the facility of the physiological aspect.
Okay. And your findings are that you can extend the shelf life by?
By up to 6 days, plus 6 days. Normally it is in good shape only up to 2 days, but we could enhance it for another 6 days.
Have you not counted decay count or microbial infestation?
We have taken the decay percentage,
Yes.
but I have not presented here. But we have noted those also because we have attempted shelf life in other indigenous fruits also.
Okay. So what is the decay percent, and what will be the endpoint of shelf life in terms of decay percent in the specific fruit?
Decay percentage is like not more than 10%.
Isn't it too low to mention? Not more than 10%. I think it is too low. And what was your sample space in the following experiment?
Our sample we have done in big lots also. We have done in small lots also. Our small lots were like 1 kg in punnets, and our big lots were like more than 10 kg in one lot.
Okay, can you please tell me how the maturity of the said fruit was judged? Based on what?
First thing is the color change. The color change.
Okay.
Because uniform color change, and also the size.
Okay. Okay, ma'am.
So, madam, you have given like guargum, isn't it? So,
yes, ma'am.
Yeah. Is it in general use for the previously, or it has been done only for this particular study?
Guargum is a plant-based extract, so it is commonly used for edible post-harvest coatings of fruits.
Yeah,
Thus
yeah,
it
edible,
improves,
it's edible, but I was saying whether it was done for this particular study or it was already used as a
in literature, it is already in use, so we have referred to literature and yeah.
Is it for the first time you are using it or it was already in use?
This fruit is first attempt. As I mentioned earlier, the post-harvest studies of these indigenous fruits is very limited. So for this particular fruit, I have not seen any literature of guar gum. So I can say in this region, this is the first attempt.
Ma'am, do you think during transportation also this will work?
Yes, ma'am.
Yeah, I think if there are no other queries, the next participant could be ready for the presentation, please.
ओके थैंक यू मैम नेक्स्ट पार्टिसिपेंट्स के लिए बोलिए रेडी हैं
Dr. Sushil Kumar, are you there? Dr. Sushil?
Yes, I am ready.
Yeah, you can share your screen, sir. I make you co-host already.
In slideshow, it is not done actually. I am trying my best.
The previous one should have been shut down and you should have been given the permission, sir, to share your screen. That is also one of the things.
The screen is shared, but it is not in the. It is in the. It is not in the actually. Now it is okay. Okay. Now it's okay.
You can start again so that if there is any lag down, you can. Yeah, it is visible, sir.
Yes.
Now it is visible.
It's visible. I am Dr. Sushil Kumar from Navsari Agriculture University, Gujarat. Today I'll discuss some of the latest techniques which have come up in the pest management or in the IPM program. Till now, apart from the chemical control, we emphasized on the biological control or the microbial pesticide, and lately on the pest behavior modifying chemicals. The genetic manipulation of pest population came very lately, and the plant immunization through other techniques other than the breeding or genetic engineering have also come up. But these are the solo efforts to modernize the IPM, not the collective efforts. And because of this, this is the actually what I want to discuss. If you sum up all the methods, then it becomes the part and parcel of the IPM. But what happened? The Stringberg in 2017. He discussed the total approach, that is the holistic approach, and that approach included the interdisciplinary approach. Interdisciplinary approach means, for example, the pest management recommendation have come from the entomologist. So that recommendation should also incorporate the recommendation of agronomist, plant breeder, agriculture engineer, the economist, collectively also. And when the collective capsule or the becomes ready, then it should be transferred to the farmer rather than the lone recommendation given by entomologist or plant pathologist for integrated disease management. So that should not be there. Steenberg emphasized on the interdisciplinary approach. Now, what can be the interdisciplinary approach nowadays should include the level of education of the farmer, economic and social condition, whether the farmer is the big farmer or small farmer or marginal farmer. What is his awareness regarding environment? What are the moral values, regulatory aspects of the government policies, and what are the available IPM tools? The extension education, the consumer preference. Extension education does not mean the transfer of technology. Extension education means what is the need of the farmer should be determined by the extension education specialist. It is before the formulation of the IPM, not after the formulation of the IPM. and the retail. So there is no at present IPM model which encompasses all these methods, all these factors. Now, in biological control, for example, we are using the natural enemies in the form of predators, parasitoids, microbial agents, but they are or they have been effective till date on a large, on a small scale, or maybe in greenhouse, but not in the form of area-wide management. In the area-wide management, the biological control, including the consisting of natural enemies, has not been very effective. Now, the farmers with greater income, means the big farmers, they can manipulate the recommendation in their favor, and the small farmer, the marginal farmer, sometimes he is not aware. Now, some people think that the IPM is a rotation of the chemicals having different mode of action, but it is untrue. It may be true, but it is not true completely. Then host plant resistance, that the use of tolerant variety should be there. Then the pheromones, that means the behavior changing chemicals in the form of mating disruption, mass trapping, monitoring of certain pests, is there, but not of all. For example, for hemipteran pests, sucking pests, or thrips, no such pheromone technology is currently available. Then mechanical tools, they may be effective for high value crops like strawberry, but not in non-specialty crops. For example, in cereals, millets. Nowadays, the government is emphasizing on the millets. There is no such mechanical tool which can be used in those crops. Then, if there is area-wide management need, then the chemical control is always the first line of action. Okay, seed control, maybe seed treatment may be effective, but not in all the cases. In horticulture crop, it's not so effective. Then the molting hormone, or the chemical which mimics, that means the analogs, the chitin synthesis inhibitors, or the JH, that is juvenile hormones. They are the recent examples, and which have been added in the recent IPM modules. Now, besides the recent which I have discussed, the knowledge of the grower, resource of the grower, planning and organization of should also be there. Now, these are the first. In pest management, the crop attributes should be there. As I have discussed, the cultural control, that means the agronomic component of the IPM should always be an important component. Then the behavior, that means the pheromones or JH analogs. Then the mechanical control, the microbial control, and the therapeutics. What is therapeutics? This word has come very recently in IPM. The pathogens or parasitoids, which we are rearing in the lab or in the field, are available and are being touted as therapeutics. That means they should not be the last option. They should be the first or the second option. Now, here you see the pest and the resource. Resource means the planting material or the plant. Now, they are manipulating the behavior of the pest, and behavior within the same population, that means of the same species, which is known as interspecific, or between the two species of insects or insect pests. Then netting. Netting is generally used in horticulture crops, particularly in the high value fruit crops. In the third, you see, in the case of apple, the column cages are used particularly against the fruit fly. Now, in the new method, the biorational products. What are biorational? That I will discuss in the next slide. IGR, heat treatments, vapor heat treatment. They are used particularly in case of high value food crops. Then biotechnology, that is the gene editing technology, CRISPR method. And the biotechnology means the use of genetically engineered plants. So these are the new technologies, although they have been developed solely. But if we combine all in one formulation, then it becomes a new IPM model rather than the old model. Now, biorational products means maybe the botanicals of flora, or natural toxins like the nicotine alkaloids or neem products. Now, what are the IGR components? IGR component, for example, pyriproxyphene. The pyriproxyphene reduces the egg viability. Egg viability means the hatching percentage. Then it also reduces the sex determination. Then there are two more chemicals as I share, like one is diflubenzuron and another is metoprene. This keeps the larva in the stage. That means it doesn't allow, it increases the molting period. It doesn't allow the larva to convert it into the pupa. Then pyriproxyfen also results in the large fat larvae. Then cryomazine. Cryomazine is another IGR which keeps the larva in molting. That means it lengthens the molting period or the metamorphosis of the period. Then in case of pupa, pyroxyfen causes the pupal motility or the pre-pupal motility. So these are the components, may not be as fast as the toxic chemical pesticide, but they are definitely an important component of the IPM. Then vapor heat treatment. Vapor heat treatment I have said is mostly in case of fruit fly. Fruit fly of different subtropical fruits as well as apple temperate fruit. And then stone weevil, a very important pest in mango. Then seed borer. Seed borer is actually multi-species pest. In case of sapota, it is a very important, that is a Trimeltis margarius. So in all this, the vapor heat treatment has been found very effective, and this treatment does not use any chemical, only using a particular temperature and particular humidity for killing the inside maggot or inside the larva, or doesn't allow even the eggs to hatch. Then then CRISPR technology, gene editing technology. There are different genes, and phenotype related genes, female related genes like the X-specific, metabolism related genes, segmentation related genes, pheromones and other related genes, insecticide resistance related genes. So these genes are manipulated, edited using the CRISPR technology, and now using this CRISPR technology, we can incorporate one more component in the IPM, in the new IPM. This is a diagrammatic view of the CRISPR technology. Then second generation green products. Second generation green products are different from the green products which we are using nowadays. Nowadays, the botanicals have a very unpleasant smell or odor. Because of this, in case of high value, in case of those crops which are harvested very frequently at a very short interval, these botanicals are not preferred by the farmer. So the second generation green products which have a good aroma, good pleasant smell, they are generally used. Then rodent birth control is controlled. Another than fly baits. Fly baits is generally used in the form of stickers near the food storage area where the flies congregate. That means they are coated with the insect food and insecticide. Insecticide means a safer insecticide, and that knocks down the landing of the female adult for egg laying. Then socio-ecological tools. Socio-ecological tools means the awareness of the farmer, environmental awareness, economic condition of the farmer. Then his knowledge regarding the implementation of the IPM model, IPM model, that should also be incorporated. We cannot have one IPM model for all types of farmers. It has to be crop wise, it has to be farmer wise, it has to be the area wise, and it has to be the region and agro ecosystem wise. We cannot, because the problem is why the IPM model developed for different crops in different regions, they do not work after 3 to 4 years. The reason is we are not considering or discussing the socio-ecological aspect of the farmer. We are in fact neglecting this aspect, and this aspect is very, very important. Firstly. Secondly, in market, there are different players. Player number one is ICR or agriculture university. Okay.
Because the time is lagging.
Then the player number two is agrochemical industry. They have their own set of recommendations, and the farmer is confused which recommendation to choose from or which is not to choose. So this SES model should be important. Okay.
But, still, how many slides? Okay. Okay.
This is the last slide.
Okay.
So the earlier IPM model focused on the ecological or environmental aspect, but now the time has come to incorporate all the available disciplines, all the socio-ecological aspects of the farmer, of the area, and encapsulate in one strategy. So that is the new IPM model which we are going to discuss and which we are going to implement for crop-wise, area-wise, farmer-wise, agro-ecosystem-wise. Thank you very much.
And it was a good focus on the IPM, sir.
Yes.
But how far can we rely only on the what you say biological methods or the physical methods, sir?
See what happens. Earlier, we recommended the biological methods, but biological methods were alone used in area-wide management. Whenever there is a very harmful pest, like for example Helicoverpa, for example Spotoptera frugipata recently, or some species of thrips which have recently come in during the last 1 or 2 years. You cannot have the biological control method. You have to incorporate all these methods in one capsule. And secondly, the recommendation should not be the uniform. It should be area-wise. It should be farmer-wise. You give a separate set of recommendation for small and marginal farmer. Give a separate set of recommendation for big farmers who has no money problem, who can implement or spend any money in as per his own wealth. But that method, that theory is a big failure in case of small and marginal farmers. Remember, in India, more than 75% farmers are small and marginal. You cannot overlook them.
Nice presentation, sir. Can we move on to the next presentation, please? If there are no queries from the participants.
Thank you very much.
Thank you.
Okay.
रोहित जी नेक्स्ट के लिए बोलिए.
That's it.
Is my screen visible?
Yeah, it
Haji.
is visible, yes, visible.
So a very good afternoon to one and all. I'm Mr. Tuswing Alphonse Haumai, a PhD scholar at ICR National Dairy Research Institute, Karnal. My topic for today's discussion is response of wheat yields to meteorological drought in northwest India. So India is the world's second largest wheat producer after China. So by drought, it means a region experiencing persistently below average precipitation. So drought is classified into 4 types, namely hydrological drought, meteorological drought, agricultural drought, and socio-economic drought. And among the many different types of drought, meteorological drought is of utmost importance since surface water resources are strongly reliant on meteorological information. And with temperatures expected to rise over the next few years, yields in many wheat producing regions are expected to fall. Punjab and Haryana are important wheat producing states in the country. So the objective of the study is to analyze the relationship of meteorological drought with yield of the crop. Coming to the material and methodology, firstly, it is standardized precipitation evaporation index, which is based on rainfall and temperature. The next is standardized yield residual series, SYRS. And the yield of the crop does not depend only on rainfall and temperature, but it also depends on other factors like agricultural technology, irrigation, fertilizer application. So in order to remove the bias caused by non-climatic factors, using simple linear regression model, the yield data were detrended. The next methods is the Spearman's rank correlation test. The study area is Punjab and Haryana. Basically, this is a panel data from the year 1966 to 2017. Coming to the results and discussion. So first of all, let me tell what SPEI 1 and SPEI 5 means. So SPEI 1, which means drought occurring particularly in 1 month. Say, for example, SPEI 1 November, which means drought occurring in the month of November. And SPEI 5 March, which means the drought occurring for the last 5 months. That is from November to March, which is considered as a growing period for wheat crop. And from the table, we can see that there is a significant positive correlation between Amritsar SPEI 1 February, Hoshiarpur SPEI 1 November, Ludhiana SPEI 1 November, March, and SPEI 5 March, and a negative significant correlation between SPEI in Patiala between SPEI 1 March. Whereas if we look in the state of Haryana, there is a significant positive correlation only in the district of Ambala, SPI 1 in the month of January. So coming to the figure, the scatter plot of SYRS width and the SPI yield. So there is a correlation between SYRS and SPI 5 March, though the correlation is weak. Now coming to the conclusion, the study shows there is a positive correlation in Hoshiarpur, Ludhiana, Amritsar, and Ambala in SPI 1, whereas in SPI 5 March, that is a positive correlation in Amritsar and Ludhiana is observed. So coming to the policy implications, with the irrigation facilities greatly developed, these two states are considered generally resilient to drought. However, with the unsustainable use of water resources, the main source of irrigation in the state can affect climate change and reduce the productivity of crops. Hence, policies like the rainwater harvesting, widening extension department views, and also developing drought stress tolerant varieties of seeds can assist in mitigating the impacts of climate change in up to certain extent. Thank you.
रोहित जी, कितने सेशंस और कितने लोगों के और बचे हैं, जरा बताएंगे एक बार देख के।
7 सर।
कितने?
7 पार्टिसिपेंट।
ठीक है, तो इसके बाद में फिर ब्रेक कर देते हैं, 3 बजे के लगभग में आएंगे, क्योंकि लंच ब्रेक भी देना है, 40 मिनट वैसे ही एक्स्ट्रा हो गया है।
जी सर। अभी
मैडम भानुरेखा, प्लीज क्वेश्चंस आस्क करिए आप।
I hope Alphonse.
Yes, ma'am. Yes, ma'am.
Yeah. So, how do you think that this model will be useful? Is it applicable for other wheat areas? I think it is a simple regression model.
Ma'am, this is a correlation.
So, what was the correlation extent that you could see for this particular model?
Actually, the correlation is not very strong, but
That's what I was asking.
the correlation is weak. However, there is the weak correlation we can observe.
Okay. So that means it is not really applicable to the extent.
Actually, meteorological drought, it always occurs. So it is applicable with the changing of climates. It will be a great impact.
Parameter, some other type of analysis could have been also done because the correlation is very less. So this correlation being very less, I don't know how far it is applicable. So is there any other way out for having a better interpretation?
Ma'am, I can also include the prices,
Yes.
the variables of price.
But when the correlation is less, I just have a doubt, what is the response that you could get? On what thing we could conclude? Other way of interpretation could have been also given as an alternate way because it is a weak correlation.
Okay, ma'am.
Just on that we cannot conclude, isn't it? When it is not so strong correlation.
Okay.
Oh.
मैडम भानुरेखा, क्या हम लंच ब्रेक ले सकते हैं, फिर 3 बजे के बाद स्टार्ट करें?
Yes, sir. It will be difficult because there may be many participants.
जी, किरण मैडम और हमारे जो HAU की टीम है, मैं आदेश लेना चाहूंगा कि क्या हम लंच के लिए ब्रेक ले सकते हैं?
जी सर, ले सकते हैं।
Exactly, we will join by 3, sir, so that we can wind up the remaining things.
हाँ, remaining. जी मैडम।
I also have to present two oral presentations, sir.
Yes, ma'am. Yes, ma'am. Please. Yes.
Last, I will try to present. We will join exactly sharply by 3, sir.
Okay, madam. Okay, madam.
Thank you. Thank
you. Thank you, madam. Okay.
Thank you.
Lunch break.